The genetics of cancer. The Hayflick limit, also known as the Hayflick phenomenon, refers to the number of times a normal somatic or differentiated human cell population will divide before cell division stops. This concept was introduced by American anatomist Leonard Hayflick in 1961. He demonstrated that normal human fetal cells in cell culture divide approximately 40 to 60 times before entering a senescence phase. Senescence in this context is where cell replication rate slows before halting altogether. This finding actually disproved the earlier claim made by Alexis Carrel that normal cells are immortal. McFarlane Burnett later coined the term Hayflick limit in his 1974 book Intrinsic Mutagenesis, a genetic approach to aging. Hayflick interpreted his discovery as aging at the cellular level, suggesting that the aging of cell populations correlates with the overall physical aging of an organism. Now, let's talk about telomeres. During DNA replication, small segments of DNA within each telomere, which are located at the ends of chromosomes, cannot be copied and are lost. Telomeres are non-coding regions of DNA, meaning they do not code for any proteins. They consist of a repeated code on the end region of linear eukaryotic chromosomes. After numerous divisions, the telomere is shortened to a critical length, and the cell becomes senescent, reaching its Hayflick limit. Interestingly, Hayflick discovered that only cancer cells are immortal. Cellular senescence doesn't occur in most cancer cells because of an enzyme called telomerase. Telomerase extends telomeres, preventing them from shortening in cancer cells, thus giving them infinite replicative potential. Telomerase is expressed activated and replaces the missing pieces of telomeres. Telomeres are composed of 4 chemicals, and these 4 chemicals are reattached to the DNA molecule at each round of division, helping to maintain telomere length more or less constant. Some telomeres are lost, but they were never lost to the extent in cancer cells as they were lost in normal cells. Given this, a proposed cancer treatment involves using telomerase inhibitors to prevent telomere restoration, allowing the cancer cells to die like normal body cells. In short, the Hayflick limit describes the finite number of divisions a normal cell can undergo primarily due to telomere shortening, while cancer cells often bypass this limit through the action of telomerase, granting them immortality. Replicative senescence is a state where cells lose their ability to divide after a certain number of divisions, typically due to the shortening of telomeres. Telomeres are protective caps at the ends of chromosomes that shorten with each cell division. Once they reach a critical length, the cell triggers a series of responses that lead to permanent growth, arrest, meaning the cell can no longer replicate. This process acts as a natural barrier against uncontrolled cell proliferation, which is a hallmark of cancer. In normal cells, replicative senescence serves as a crucial tumor suppressive mechanism. When normal somatic cells undergo stress, like DNA damage or telomere shortening, they enter this senescent state, which helps prevent the potential development of tumors. The activation of tumor suppressor proteins such as 53 and 16-amphora plays a significant role in this process. 53 is a well-known tumor suppressor that responds to cellular stress by inducing cell cycle arrest, allowing time for repair, or leading to apoptosis if damage is irreparable. Conversely, in cancer cells, the scenario is quite different. Cancer cells often bypass replicative senescence through various mechanisms, including the reactivation of telomerase, an enzyme that extends telomerase. This allows cancer cells to continue dividing indefinitely, a phenomenon known as replicative immortality. The inactivation of 53 and 16-amphora pathways in cancer cells removes the checks and balances that would normally trigger senescence. As a result, these cells can proliferate uncontrollably. Now, let's discuss crisis, which refers to a stage following replicative senescence, where cells experience extreme genomic instability and undergo massive cell death. In normal cells, Crisis occurs when telomeres become critically short and can no longer maintain chromosome integrity. This leads to catastrophic events like chromosomal fusions and further DNA damage. In cancer cells, crisis can also occur, but is often bypassed due to mutations or viral infections that allow them to escape this fate. For instance, some cancer cells may express viral proteins that mimic or inhibit cellular pathways involved in senescence. If these cancer cells manage to reactivate telomerase or other pathways that promote survival, despite crisis conditions, they can transition from crisis back into a state of active proliferation. To summarize, replicative senescence in normal cells, induced by telomere shortening, serves as a protective mechanism against tumor formation, involves activation of tumor suppressors like 53 and 16-4A. Replicative senescence in cancer cells, often bypassed due to mutations or activation of telomerase, leads to replicative immortality. Lack of functional tumor suppressors allows uncontrolled division. Crisis in normal cells occurs after prolonged replicative senescence, characterized by genomic instability and cell death. Crisis in cancer cells can lead to massive cell death, but may be evaded through mutations or viral mechanisms. Successful evasion can lead back to active proliferation. Let's dive into the end replication problem and how it affects telomeres. The end replication problem arises because of how DNA replication works in linear chromosomes, which are found in eukaryotic cells. Unlike circular chromosomes in bacteria, linear chromosomes have ends, and these ends can't be fully copied during DNA replication. Here's the breakdown. During DNA replication, the DNA double helix is unwound, and each strand serves as a template for a new strand. One of the new strands, called the leading strand, is synthesized continuously in the 5 to 3 direction. The other strand, called lagging strand, is synthesized in short fragments called Okazaki fragments. Each Okazaki fragment requires an RNA primer to initiate DNA synthesis. These RNA primers are later removed and replaced with DNA. The problem occurs at the very end of the chromosome during lagging strand synthesis. After the RNA primer is removed from the last Okazaki fragment, there's no adjacent 3O group available for DNA polymerase to extend and fill the gap with DNA. This results in a small stretch of single-stranded DNA at the end of the chromosome. Because DNA polymerase can only add nucleotides to the 3 end of an existing nucleotide chain, the extreme 5 end of a DNA strand cannot be completely copied in each round of replication. Consequently, part of the DNA at the end of a eukaryotic chromosome goes uncopied in each round of replication, leaving a single-stranded overhang. Now, how does this relate to telomerase? Telomeres are repetitive nucleotide sequences located at the ends of linear chromosomes. In humans, this sequence is typically TAG. Telomeres protect the ends of chromosomes from degradation and prevent them from fusing with neighboring chromosomes. They act like protective caps, ensuring the integrity of the genome. Because of the end replication problem, telomeres shorten with each round of DNA replication. If telomeres become too short, they can no longer protect the chromosome ends, leading to DNA damage, genomic instability, cellular senescence, aging, and potentially apoptosis, programmed cell death. Interestingly, recent research indicates there may actually be 2 end replication problems. The first well-known problem relates to the incomplete copying of the leading strand, which telomerase addresses by adding T-tag repeats to the telomere end. The second problem involves incomplete duplication of the C-rich telomeric repeat strand during lagging strand synthesis. The CST polyprimase complex helps to resolve this second issue. In summary, the end replication problem leads to the gradual shortening of telomeres with each cell division. This shortening can eventually compromise chromosome stability and cell Viability. The enzyme telomerase can counteract this shortening by adding telomeric repeats, but it is not active in most somatic cells. Let's revisit the end replication problem and its impact on telomeres incorporating recent findings. The end replication problem arises due to the nature of linear DNA replication in eukaryotic chromosomes. Unlike circular chromosomes, linear chromosomes have ends that pose a challenge for complete replication. During DNA replication, the DNA double helix unwinds, and each strand serves as a template. The leading strand is synthesized continuously in the 5 to 3 direction. The lagging strand is synthesized discontinuously in short fragments called Okazaki fragments, each requiring an RNA primer. After RNA primers are removed, DNA polymerase fills the gaps, but at the chromosome's end, the final RNA primer on the lagging strand can't be replaced with DNA because there no upstream 3O group for DNA polymerase to extend from. This leaves a single-stranded DNA at the chromosome's end. Telomeres are repetitive nucleotide sequences, tag in humans, at the ends of linear chromosomes, protecting them from degradation and fusion. The end replication problem causes telomeres to shorten with each DNA replication round. If telomeres shorten critically, chromosome stability is compromised, leading to senescence or apoptosis. Now, here's where it gets interesting with the recent research. It turns out there are potentially two end replication problems. One, the first end replication problem, the classical one, relates to the incomplete copying of the leading strand. Telomerase, a reverse transcriptase enzyme, addresses this by adding G-rich telomeric repeats to the three ends of telomerase. This counteracts telomere shortening caused by the loss of telomeric 3 overhangs during leading strand DNA synthesis. 2. The second end replication problem involves the incomplete duplication of the C-rich telomeric repeat strand during lagging strand synthesis. The CSD poloaprimase complex helps solve this issue. In vitro studies show that priming for lagging strand DNA replication doesn't occur on the 3 overhang, and lagging strand synthesis stops 150 nucleotides away from the template's end. Without CST polyoprymase, lagging end telomeres lose 50-60 nucleotides of CCTA repeats per population, doubling. In essence, canonical DNA replication creates two telomere end replication problems. Telomerase maintains the G strand, and CST polyoprymase maintains the C strand. The leading end telomeres shorten by approximately 100 ntpd, reflecting the generation of 3 overhangs through resection. Therefore, the end replication problem isn't just one issue but a dual challenge that requires telomerase and the CST polyoprymase complex to ensure complete telomere replication and genome stability. Let's break down how telomerase enables cancer cells to achieve immortality. In normal somatic cells, telomeres shorten with each cell division due to the end replication problem. Once telomeres reach a critically short length, they signal the cell to stop dividing and enter senescence or undergo apoptosis. This mechanism limits the number of divisions a normal cell can undergo, preventing uncontrolled proliferation and tumor formation. Cancer cells, however, often bypass this limitation by reactivating telomerase. Telomerase is a reverse transcriptase enzyme that adds repetitive DNA sequences, tag in humans, to the ends of telomerase, counteracting the shortening that occurs during DNA replication. By maintaining telomere length, telomerase prevents cancer cells from reaching the critical telomere length that would normally trigger senescence or apoptosis. This allows cancer cells to divide indefinitely, achieving replicative immortality, a hallmark of cancer. Here is a more detailed breakdown. 1. Telomere shortening in normal cells. Normal cells have a limited number of cell divisions because telomeres shorten with each division. 2. Critical telomere length. Once telomeres become critically short, They trigger DNA damage responses leading to cell cycle arrest senescence or apoptosis. 3. Telomerase reactivation in cancer cells. Cancer cells reactivate telomerase, which adds telomeric repeats to chromosome ends, maintaining telomere length. 4. Replicative immortality. By preventing telomere shortening, telomerase allows cancer cells to bypass the normal limits on cell division and proliferate indefinitely. It's important to note that telomerase is not active in most normal somatic cells, which is why telomeres shorten over time in these cells. However, telomerase is active in germ cells and stem cells, ensuring that these cells can divide repeatedly without telomere shortening. Interestingly, some cancer cells use an alternative mechanism called ALT, alternative lengthening of telomeres, to maintain telomere length without telomerase. ALT relies on DNA repair mechanisms to elongate telomeres, but it's less common than telomerase reactivation. Given telomerase's crucial role in cancer cell immortality, it has become a target for cancer therapy. Several strategies are being developed to inhibit telomerase activity with the goal of causing telomere shortening in cancer cells, ultimately leading to senescence and apoptosis. One such drug, imetelstate, directly inhibits telomerase activity and has been approved for treating certain hematological malignancies. Let's delve into the alternative lengthening of telomeres, mechanism, and its role in telomerase-negative cancers. First, remember that normal cells rely on telomerase to maintain telomere length, thus avoiding senescence. However, most somatic cells do not have active telomerase. Cancer cells need to overcome this, and while many achieve this by reactivating telomerase, a subset uses an alternative pathway called ALT. The ALT mechanism is a telomerase-independent way for cancer cells to maintain their telomeres, gaining replicative immortality. It's particularly important in cancers that do not express telomerase. ALT is found in about 5–10% of all cancers, but it's more common in certain types like sarcomas, astrocytomas, pancreatic neuroendocrine, tumors neuroblastoma, and chromophobe hepatocellular carcinomas. So how does ALT work? ALT relies on homology-directed repair, a type of DNA repair to elongate telomeres. Specifically, it uses a process called break-induced replication, BR. This resembles the way broken DNA strands are usually repaired with a matching undamaged DNA sequence. But in bits, this process is used to extend telomerase. Telomeres are repetitive, so matching sequences are readily available. Here's a step-by-step breakdown based on proposed models. 1. Telomere damage. The process starts with damage to a telomere, possibly a double-stranded DNA break, chronic replication, stress, and/or telomere erosion. 2. Resection. One of the DNA strands is cut away, resected, creating a single-stranded DNA, the guanosine-rich strand. 3. D-loop formation. This single-stranded DNA invades a template sequence, forming a displacement loop, D-loop. The template can be a centromere proximal sequence of the same chromosome, T-loop. 3. Extrachromosomal telomeric sequences, C-circles, homologous chromosomes, other chromosomes. 4. DNA synthesis. DNA polymerase extends the invaded G-strand copying material beyond the original breakpoint. Lagging strand synthesis of the C-strand also occurs. 5. Non-conservative DNA product. Both strands end up containing entirely new DNA, unlike normal DNA replication, where one strand is newly synthesized and the other is from the original template. ALT also involves unique molecular characteristics. ALT-associated PML bodies, APBs, large ultra-bright nuclear telomeric foci, partially single-stranded telomeric DNA circles, C-circles, telomeric long non-coding RNA, TERA. ALT can lead to widespread chromosomal instability, chronic replication stress, and telomere length heterogeneity. Also, alt-positive cancers often have mutations in chromatin, remodeling proteins, HEXDAX and H33, or DNA damage repair factors, smarcalonin and SLX4-ep. While alt allows cancer cells to maintain telomeres, it can also jeopardize telomere integrity if not controlled. Increased transcription of TERA, for instance, can increase alt activity, but also cause telomeric DNA loss. The alt mechanism has molecular events with the potential to endanger telomere stability. PC4. A multifunctional protein has been identified as a novel factor, essential for alt cell viability. Depletion of PC4 results in rapid alt cell death, induces replication stress and telomere fragility in alt cells, and increases alt activity. In summary, the alt mechanism is a crucial telomerase-independent pathway that enables certain cancer cells to maintain telomere length and achieve immortality. It involves homology-directed repair, break-induced replication, and several unique molecular characteristics. Targeting alt is being explored as a potential cancer therapy. Let's discuss how T-loops and their associated proteins work together to protect telomeres. Telomeres, as we've discussed, are repetitive DNA sequences at the ends of chromosomes that protect the genome from degradation and fusion. But telomeres themselves need protection. That's where T-loops come in. A T-loop, telomere loop, is a large loop-like structure formed at the end of a telomere. It's created when the three single-stranded overhang of the telomere invades the double-stranded DNA region of the telomere, forming a displacement loop, D-loop. D-loop. Think of it like tucking the loose end of a rope back into the rope itself to prevent fraying. This knot stabilizes the telomere and prevents it from being recognized as a DNA break by the DNA repair machinery. Without T-loops, the cell might mistake the telomere for a double-strand break and try to fix it, which could lead to chromosome fusion and genomic instability. Key to this protection is a protein complex called shelterin. Shelterin is a multi-protein complex that binds to telomeres and regulates telomere length, structure, and function. In humans, shelterin consists of 6 core proteins: TARFONE, telomeric repeat binding factor 1, and TF2, telomeric repeat binding factor 2. These bind directly to the double-stranded telomeric DNA. TINT2, TARFONE interacting, nuclear factor 2. TINT2 acts as a central hub connecting TRFONE, TF2, and other shelterin componency. POTONE, protection of telomeres 1. POTONE binds to the single-stranded 3 overhang. TIPONE, also known as ACD, it interacts with POTONE and TINT2, bridging the single-stranded and double-stranded DNA binding componency. RAPONE, repressor, activator protein 1. RAPONE binds to TRF2 and is involved in telomere length, regulation, and gene silencing. Here's how T-loops and shelterin work together to protect telomeres. Preventing DNA damage response. T-loops sequester the chromosome terminus from DNA end sensors, preventing inappropriate activation of the DNA damage response, DDR, at the ends of linear chromosomes. Inhibition of DNA repair. Shelterin inhibits key steps in the DNA damage response, preventing the telomere from being processed as a double-strand break. TRF2 in particular represses ATM signaling and non-homologous end joining. T-loop formation and dynamics. TRF2 is crucial for T-loop formation. Studies show that reducing TRF2 levels leads to a structural change from T-loops to linearized chromosome ends, which activates ATM. The TRF domain of TRF2 regulates T-loop formation while suppressing ATM activity. Shelterin complex stability. The shelterin complex maintains the T-loop structure and ensures that the telomere end is protected. For example, TIN2 binds to TARFONE and TRF2, stabilizing the complex. TERA involvement. Telomeric repeat containing RNA, TERA, can also play a role in T-loop formation and telomere protection. TERA can bind to TRF2, recruiting the shelterin complex to internal R loops within the telomere. In summary, T-loops maintained by the shelterin complex act as protective caps at the ends of chromosomes. They prevent the telomere from being recognized as damaged DNA, inhibit inappropriate DNA repair, and maintain genomic stability. Let's break down how 53 and RB, retinoblastoma protein, regulate replicative senescence. Replicative senescence, as we've discussed, is the process where cells permanently stop dividing after a certain number of divisions, largely due to telomere shortening and the end replication problem. Thus, this is a critical tumor suppressor mechanism, preventing cells with damaged DNA from proliferating uncontrollably. Both 53 and RB are key players in enforcing this cellular fate. 53's role in replicative senescence. 53, often called the guardian of the genome, is a transcription factor activated by various cellular stresses, including DNA homogeny activation, hypoxia, metabolic dysfunction, and replicative stress. When activated, 53 initiates several responses to prevent cancer initiation. One of these responses is the induction of senescence. Here is how 53 induces senescence. First, activation by telomere shortening. As telomeres shorten with each cell division, they eventually trigger a DNA damage response. This activates 53. Second, transcriptional activation. Activated 53 functions as a transcription factor, binding to DNA and promoting the expression of target genes. Key target genes include 1. P21, CD-canonia. P21 is a cyclin-dependent kinase inhibitor, CDKI. It inhibits cyclin-dependent kinases, CDKs, which are enzymes that drive the cell cycle. By inhibiting CDKs, P21 blocks the cell cycle progression, particularly at the GON-S and G2M checkpoints. 2. BAX and PUMA. 53 can induce apoptosis through BAX and PUMA activation. 3. Long non-coding RNA neatone. Induction of neatone expression contributes to the tumor suppressor function of 53. Third, cell cycle arrest and senescence. The upregulation of P21 leads to cell cycle arrest. If the DNA damage is irreparable or the stress persists, 53 can drive the cell into permanent senescence. RB's role in replicative senescence. The retinoblastoma protein, RB, is another critical tumor suppressor that regulates cell cycle progression. Unlike 53, which is activated by stress signals, RB functions continuously in the cell cycle. Here's how RB induces senescence. 1. Hypophosphorylated state. In its active state, RB is hypophosphorylated, meaning it has few phosphate groups attached. In this state, RB binds to and inhibits ETWF transcription factors. ETWFs are crucial for the expression of genes required for DNA replication and cell cycle progression. 2. Cell cycle arrest. By inhibiting ETWFs, RB prevents cells from entering S phase, DNA replication, and progressing through the cell cycle. 3. Senescence reinforcement. During senescence, RB remains in its hypophosphorylated active state, ensuring that ETWF target genes remain repressed, thus reinforcing the senescent state. Synergistic action. 53 and RB often work together to enforce senescence. For example, P21, a target of 53, inhibits CDKs, which normally phosphorylate and inactivate RB. By inhibiting CDKs, P21 helps maintain RB in its active hypophosphorylated state, further suppressing ETWF activity and reinforcing the senescent state. In summary, 53 and RB are critical regulators of replicative senescence. 53 responds to cellular stresses by upregulating genes like P21, leading to cell cycle arrest and senescence. ORB, in its active state, inhibits ETWF transcription factors, preventing cell cycle progression and reinforcing senescence. Together, they ensure that cells with critically shortened telomeres or other forms of irreparable damage are permanently removed from the cell cycle. Preventing tumor development. Let's discuss how the SV40 large T antigen, LTAG, contributes to cell immortalization. SUI40 ELTAG, derived from the simian virus 40, is a viral oncoprotein widely used to immortalize various cell types in research. Immortalization in this context means extending a cell's replicative lifespan, indefinitely bypassing the normal limits of cell division imposed by senescence or crisis. Here's how SUI40 ELTAG achieves this. 1. Inactivation of tumor suppressor proteins. First option, 53 inactivation. SUI40 ELTAG binds to 53, a crucial tumor suppressor protein. By binding to 53, ELTAG blocks its ability to activate transcription of its target genes. Normally, 53 responds to cellular stress, like DNA damage or telomere shortening, by inducing cell cycle arrest, apoptosis, or senescence. By inactivating 53, SUI40 ELTAG prevents these responses, allowing cells to continue dividing even when they normally wouldn't. Second option, RB inactivation. SUI40 TAG also binds to the retinoblastoma protein, RB, another key tumor suppressor. RB normally inhibits ETWF transcription factors, which are essential for cell cycle progression. When RB is bound to TAG, it can no longer inhibit ETWFs, leading to increased expression of genes required for DNA replication and cell division. This forces the cell cycle to continue even in the presence of DNA damage. 2. Activation of ETWF-mediated transcription. By binding with RB, ETWF complex SV40 ELTAG activates ETWF-mediated transcription, further promoting cell proliferation. 3. Telomerase activation. Recent studies indicate that SV40 ELTAG can induce telomerase activity in infected cells. Telomerase, as we've covered, is an enzyme that maintains telomere length, preventing telomere shortening and replicative senescence. 4. Overcoming growth arrest and apoptosis. Through the inactivation of 53 and RB, SUI40 TAG helps cells overcome growth arrest and prevents apoptosis, resulting in sustained cellular proliferation. 5. Two-step mechanism. CV40 induced immortalization often proceeds in 2 phases. First option, lifespan extension. In the first phase, ELTAG extends the cell's lifespan by binding to 53 and RB, allowing it to continue dividing beyond its normal limit. Second option, immortalization. The second phase, which occurs in a small minority of cells, involves a gene inactivation event that leads to indefinite growth. The exact genes involved are not fully known. In summary, SV40 ELTAG promotes cell immortalization by inactivating key tumor suppressor proteins 53 and RB, activating ETWF transcription factors, potentially inducing telomerase activity, and preventing growth arrest and apoptosis. It's a powerful tool for generating immortalized cell lines in research, but it's important to remember that these cells may have altered characteristics compared to normal cells due to the effects of SV40 ELTAG. Let's discuss why mouse models have limitations when it comes to studying telomere-driven cancer, particularly in the context of human biology. The primary reason mouse models are limited in studying telomere-driven cancer is the vast difference in telomere length between mice and humans. Mouse telomeres are significantly longer, 2100 kb, than human telomeres, 515 kb. This means that mice can tolerate telomere dysfunction much better than humans. Here's the breakdown. 1. Telomere length and cancer development. In humans, short telomeres are associated with increased genomic instability and can significantly impact cancer progression. Dysfunctional telomeres can induce chromosomal instability, which may either promote or impede tumor cell proliferation, depending on other factors. 2. Telomere dysfunction and tumorigenesis. Telomere attrition can promote genome instability, potentially stimulating the initiation of early-stage cancers. However, it can also inhibit tumorigenesis by promoting permanent cell growth arrest or death. 3. Mouse models and telomerase. Mouse models constitutively lacking telomerase may not accurately reflect how telomere losses within telomerase-proficient cells impact carcinogenesis. Human cancers likely develop in cell lineages with low levels of telomerase, leading to telomere losses in early lesions, followed by subsequent activation of telomerase. 4. Hut mice. To address these limitations, researchers have developed hut, human telomere, mice which have telomeres closer in length to those of humans. These mice are expected to serve as a better model for studying tumorigenesis and age-related diseases, offering valuable insights into cancer development and potential therapies. 5. Immune system and telomeres. Short telomeres can lead to immune senescence, which may contribute to higher tumor incidences. Host T-cell immune deficiency, rather than tumor cell chromosome instability, may predispose individuals with short telomere syndromes to squamous cancers. 6. Chemotherapy and telomeres. Chemotherapy drugs can induce DNA damage in both normal and tumor cells. Tissues and cells with short telomeres are inherently more susceptible to such damage. This vulnerability could synergize with the cytotoxic effects of cancer drugs, making hut mice a more accurate preclinical model for evaluating drug toxicity. In summary, the significant difference in telomere length between mice and humans makes it challenging to accurately model telomere-driven cancer in standard mouse models. Hut mice with human-like telomere lengths offer a promising alternative for studying tumorigenesis, age-related diseases, and drug toxicity. Let's discuss the potential and challenges of targeting telomerase in cancer therapy. The potential. Telomerase, as we know, is an enzyme that maintains telomere length, allowing cancer cells to divide indefinitely. Because telomerase is active in the vast majority of cancer cells, over 85% and up to 100% in adenocarcinomas, including breast cancer, but is typically inactive in most cancer. Adult somatic cells. It represents an attractive target for cancer therapy. Targeting telomerase offers the potential for highly selective cancer therapeutics. If we can effectively inhibit telomerase, we can potentially limit cancer cell proliferation and induce senescence or apoptosis. Here are some of the potential therapeutic approaches. 1. Telomerase inhibitors. These are small molecules or other agents that directly inhibit telomerase activity. 2. Oligonucleotide-based therapies. These therapies use oligonucleotides, short DNA or RNA sequences, to target and inhibit telomerase subunits, such as human telomerase reverse transcriptase, HTRT, and human telomerase RNA gene, TRTRC. Examples include antisense oligonucleotides like GRON163L and T-oligos. 3. MicroRNA-based therapies. MicroRNAs, miRNAs, can be used to regulate the expression of telomerase subunits. Some miRNAs can downregulate ATRT, while others can upregulate IT. Targeting these miRNAs can help reduce telomerase activity in cancer cells. 4. G quadruplex stabilizers. These molecules, like telomestatin, can stabilize the structure of telomeric DNA, inhibiting to the telomere. 5. Immunotherapies. Telomerase-derived peptides can be used to develop cancer vaccines, that stimulate an immune response against cancer cells expressing telomerase. This approach aims to induce T cells to target and kill cancer cells. 6 combination therapies. Combining telomerase inhibitors with other cancer treatments like chemotherapy or radiation therapy may enhance their effectiveness. 7. Targeting TERT promoter mutations. Some cancers have mutations in the TERT promoter region driving increased TERT gene expression. Therapies targeting this mechanism are being explored. 8. Telomere processing inhibition. Disrupting the maintenance of telomeres activates an anti-tumor immune response. The challenges. Despite its promise, targeting telomerase in cancer therapy faces several challenges. 1. Delayed effects. Telomerase inhibition doesn't immediately kill cancer cells. Instead, it leads to gradual telomere shortening over multiple cell divisions, eventually triggering senescence or apoptosis. This delayed effect can make it difficult to assess the efficacy of telomerase inhibitors in clinical trials. 2. Therapeutic resistance. Cancer cells can develop resistance to telomerase inhibitors through various mechanisms, such as activating alternative telomere maintenance mechanisms like alternative lengthening of telomeres. 3. Off-target effects. While telomerase is largely restricted to cancer cells, it is also active in stem cells and certain immune cells. Telomerase inhibitors could potentially affect these normal cells, leading to unwanted side effects. 4. Delivery challenges. Delivering telomerase inhibitors, specifically to cancer cells, can be challenging. Developing targeted drug delivery systems is crucial to minimize off-target effects and improve efficacy. For example, a cell-penetrating peptide, CP, derived from IDART, has been shown to selectively deliver drugs to cancer cells. 5. Clinical trial failures. Despite extensive research, there have been no positive late-phase clinical trials for telomerase-based cancer vaccine. This highlights the need for improvements in vaccine design, selection of indications, and treatment combination strategies. 6. Complexity of telomerase function. Telomerase has functions beyond telomere maintenance, some of which may be unrelated to telomerase. Inhibiting these other functions could have unintended consequences. In conclusion, targeting telomerase holds significant promise for cancer therapy. However, overcoming the challenges related to delayed effects, therapeutic resistance, off-target effects, and delivery is crucial for developing effective telomerase-directed cancer therapeutics. Future research should focus on developing more specific and potent telomerase inhibitors, identifying predictive biomarkers for treatment response, and exploring rational combination strategies to maximize therapeutic benefit. Recent advances in structural models of human telomerase may boost the prospect of clinically effective drugs. From proto-oncogenes to oncogenes, let's explore how retroviruses acquire oncogenes from host genomes, leading to cancer. Retroviruses, also known as RNA tumor viruses, have a unique way of interacting with the genome of the cells they infect. This interaction can sometimes lead to the retrovirus acquiring host genes, specifically proto-oncogenes, and turning them into viral oncogenes. 1. Retroviral infection. Retroviruses infect cells by inserting a DNA copy of their RNA genome into the host cell's DNA. This DNA copy is called a provirus-2 integration and recombination. The provirus integrates randomly into the host cell's genome. Because the integration isn't specific, the provirus can land near cellular genes. During replication, the retroviral genome can recombine with the host cell's DNA. 3. Acquisition of proto-oncogenes. If the provirus integrates near a proto-oncogene, the recombination process can lead to the retrovirus picking up a copy of the proto-oncogene. Proto-oncogenes are normal cellular genes that regulate cell growth and differentiation for formation of viral oncogenes. Once the proto-oncogene is incorporated into the retroviral genome, it becomes a viral oncogene, V-ONC. The retrovirus has essentially captured a cellular gene 5 oncogene activation. When the retrovirus infects a new cell, it introduces the V-ONC into that cell. The V-ONC is often altered or overexpressed compared to the normal proto-oncogene. This can disrupt normal cell growth and differentiation, leading to uncontrolled proliferation and cancer. A classic example is the Rous sarcoma virus, RSV. RSV contains the SRC oncogene, which originated from a cellular proto-oncogene called CSRC. The VSRC gene encodes a tyrosine kinase that drives uncontrolled cell growth. There are two main types of retroviruses based on how they cause tumors. First, acute, transforming retroviruses. These viruses contain oncogenes and can induce tumors rapidly. However, they are typically defective in replication. Second, non-acute retroviruses. These viruses do not carry oncogenes themselves, but can cause tumors by integrating near proto-oncogenes and activating them. This process usually takes longer. In addition to acquiring oncogenes, retroviruses can also influence the host genome in other ways that contribute to cancer. 1. Insertional mutagenesis. The provirus can disrupt normal cellular genes by inserting itself into the middle of them. Promoter, enhancer, insertion. The provirus contains strong promoter and enhancer sequences that can activate nearby cellular genes, including proto-oncogenes. 3. Retrocopying. Retroviruses can use their reverse transcriptase enzyme to copy host RNAs and insert them back into the genome, potentially disrupting genes or creating new regulatory elements. In summary, retroviruses can acquire oncogenes from host genomes through recombination events during their replication cycle. These oncogenes, when expressed in infected cells, can disrupt normal cell growth and differentiation, leading to cancer. Furthermore, retroviruses can influence the expression of the host genome and insert themselves in various locations, sometimes leading to cancer development. Now, let's compare the roles of MyC and ROS oncogenes in cancer building on the previous explanation of how retroviruses acquire oncogenes. Both MyC and ROS are proto-oncogenes that, when mutated or overexpressed, can become oncogenes and drive cancer development. However, they function through very different mechanisms and occupy distinct roles in the cell. The MyC oncogene have one function. MyC encodes a family of transcription factors that regulate the expression of many genes involved in cell proliferation, growth, apoptosis, differentiation, and stem cell self-renewal. Proteins bind to enhancer box sequences, E-boxes, on DNA and recruit histone acetyltransferases, HATs, which activate transcription. MyC can also act as a transcriptional repressor E2 mechanism by modifying the expression of its target genes. MyC activation results in numerous biological effects, most notably driving cell proliferation and cell growth. 3. Role in cancer. The MyC oncogene is activated in the vast majority of cancers by genetic, epigenetic, or post-translational mechanisms. MyC aberrations or upregulation of MyC-related pathways occur in most cancers. Inactivation of MyC in preclinical models can result in sustained tumor regression, a phenomenon attributed to oncogene addiction. MyC activation drives cancer progression through mechanisms involving cell-intrinsic acquisition of cancer hallmarks or dysregulation of the tumor microenvironment and host immune responses. 4. Clinical significance. MyC genes and proteins are highly relevant for treating tumors. Many therapeutic agents that directly target MyC are under development. Inactivation of sumo activating enzyme, Sionase A2, in the presence of MyC hyperactivation results in mitotic catastrophe and cell death in cancer cells, suggesting that inhibitors of SUMOylation may be a possible treatment for cancer. MyC amplification is found in several cancer types, including breast, colorectal, pancreatic, gastric, and uterine cancers. Direct suppression of MyC function with novel derivatives can induce protein degradation and perturb MyC-Max interaction. Targeting MyC effector functions in pancreatic cancer by inhibiting the ATPase Ravlon-2 has also shown promise. 5. MyC and immune evasion. MyC activation can lead to downregulation of MHC I expression on tumor cells, poor CD8 T-cell infiltration, and poor response to anti-PD-1 monotherapy. The ROS oncogene, meanwhile, EPH1 function. ROS encodes a family of small GPases that act as molecular switches in cell signaling pathways. They cycle between an inactive GDP-bound state and an active GTP-bound state. When bound to GTPP, ROS activates downstream signaling pathways that regulate cell growth, proliferation, and survival. Mechanism. ROS proteins are activated by receptor tyrosine kinases, RTKs, and other cell surface receptors. Once activated, ROS recruits and activates downstream effectors such as AAF-PI3 and RAUGs, leading to the activation of MAPK, ACT, and other signaling cascades. 3. Role in cancer. ROS oncogenes are among the most frequently mutated oncogenes in human cancers. Mutations in RA typically result in a constitutively active protein that is always bound to GTP, leading to continuous activation of downstream signaling pathways. 4. Clinical significance. ROS mutations are found in a wide range of cancers, including lung, colon, and pancreatic cancers. Targeting ROS has been a major focus of cancer drug development, so the key differences. 1. Mechanism of action. MyC is a transcription factor that regulates gene expression, while RA is a small GPase that acts as a molecular switch in cell signaling pathways. 2. Downstream effects. MyC affects a broad range of cellular processes, including cell proliferation, growth, apoptosis, and differentiation. RAS primarily affects cell growth, proliferation, and survival through the activation of downstream signaling pathways. Frequency of mutation. RA oncogenes are directly mutated in many cancers, while MyC is often overexpressed or activated through other mechanisms, such as gene amplification or chromosomal translocation. 4. Therapeutic strategies. Targeting MyC has proven challenging due to its intrinsically disordered nature, leading to the exploration of indirect strategies like inhibiting its binding partners. RAS has been directly targeted, though with limited success, until recently, with the development of KRAS G12EC inhibitors. In summary, while both MyC and RAS are important oncogenes that drive cancer development, they function through distinct mechanisms and affect different aspects of cell biology. MyC acts as a master regulator of gene expression, while RAS acts as a signaling hub that controls cell growth and survival. Both are critical targets for cancer therapy, but require different therapeutic approaches. So now let's explore how the Philadelphia chromosome activates the EBL gene, specifically in the context of chronic myelogenous leukemia, CML, and some cases of acute lymphoblastic leukemia. The Philadelphia chromosome results from a reciprocal translocation between chromosomes 9 and 22. This translocation fuses the ablone tyrosine kinase gene from chromosome 9 with the BCR, breakpoint cluster region, gene on chromosome 22. The resulting fusion gene is called a BCR ablone, and it produces the BCR abl fusion protein. Here's how the Philadelphia chromosome activates ablone. 1. Loss of regulatory control. The normal ablone gene encodes a tyrosine kinase whose activity is tightly regulated. The N-terminal cap region is an important component of the intramolecular apparatus that inhibits the kinase activity of c-Abl. In the BCR ablone fusion, the ablone gene loses its normal regulatory elements, particularly those located in its 5 region 2 BCR-mediated dimerization, oligomerization. The BCR portion of the fusion protein contains a coiled coil domain that promotes dimerization or oligomerization of BCR-A. This dimerization oligomerization leads to constitutive activation of the ABL kinase domain. The coiled coil domain is essential for the transforming properties of BCR-ABL 3 constitutive kinase activity as a result of the loss of normal regulatory control and BCR-mediated dimerization. The ABL kinase domain in the BCR-ABL fusion protein is constitutively active. This means that it is always on phosphorylating its target proteins regardless of normal cellular signals. In contrast to its proto-oncogenic counterpart, c-Abl BCR-Abl, displays constitutive tyrosine kinase activity. 4. Downstream signaling pathways. The constitutively active BCR-Abl kinase phosphorylates numerous intracellular proteins, leading to the activation of several signaling pathways that promote cell proliferation survival and reduced growth factor requirements. These pathways include PI3K-RAS and STAT5. BCR-Abl also protects cells from apoptosis by upregulating BCL. BCR-Abl inhibits apoptosis through activation of a RAS-dependent signaling pathway. The BCR-Abl fusion protein is the primary driver of CML. Its constitutive kinase activity leads to uncontrolled proliferation of myeloid cells, reduced apoptosis, and genomic instability. The importance of BCR-Abl in CML is highlighted by the success of tyrosine kinase inhibitors, KIAs, such as imatinib, nilotinib, dasatinib, and ponatinib, which specifically target the ABL kinase domain and inhibit its activity. In summary, the Philadelphia chromosome activates ablone by removing its normal regulatory controls, forcing it to dimerize, and resulting in a constitutively active kinase that drives uncontrolled cell proliferation and survival. The BCR-Abl fusion protein is the molecular hallmark of CML and a key target for therapy. Now, let's distinguish between gain-of-function mutations and overexpression as mechanisms of oncogenesis. Both gain-of-function mutations and overexpression can lead to increased activity of a protein, but they achieve this through different mechanisms and have distinct consequences. The key is that gain-of-function, GOF, mutations alter the protein itself, whereas overexpression increases the amount of a normal protein. A gain-of-function mutation is a genetic alteration that results in a new or enhanced activity of a protein. This means the protein can do something it couldn't do before, or it can do what it normally does, but much more effectively or in an unregulated manner. Mechanism: GOF mutations typically involve changes in the amino acid sequence of a protein that alter its structure and function. These mutations can affect each one enzyme activity. A mutation might increase the catalytic activity of an enzyme or make it less sensitive to regulatory signals. For example, mutations in the ditonal gene can increase its histone methyltransferase activity, promoting proliferation, migration, self-renewal, and drug resistance of lung cancer cells. 2. Protein-protein interactions. A mutation might enhance a protein's ability to interact with other proteins, leading to the activation of signaling pathways. For example, co-occurring GOUAF mutations in HER2 and HER3 can modulate HER2-HER3 activation oncogenesis, and HER2 inhibitor sensitivity. 3. Ligand binding. A mutation might increase a receptor's affinity for its ligand, leading to increased signaling, even at low ligand concentrations. Protein stability. A mutation might make a protein more stable, increasing its half-life and overall activity. A germline HIF2A mutation found in hepatocellular carcinoma, HCC, resulted in a protein resistant to degradation. Upregulating target, genus 5, binding specificity. Mutations can create new binding sites for transcription factors, resulting in the overexpression of oncogenase 6, inhibition of tumor suppressors. GOF mutations in Buthertine promote oncogenesis by disrupting the function of the tumor. Suppressor FDDUC of an qualitative change. The protein itself is altered, leading to a qualitative change in its function. Examples: Ra mutations at codons 12 or 61 result in a constitutively active protein. The Philadelphia chromosome leads to a BCR-ABL fusion. Protein with constitutive kinase activity mutant 53 can upregulate receptor protein. Tyrosine kinase A. Axel increasing cell growth and motility in lung cancer cells. Overexpression, on the other hand, involves an increase in the amount of a normal protein. The protein itself is not mutated, but it is produced at higher levels than normal. Mechanism: overexpression can result from gene amplification. The gene is copied multiple times, leading to increased RNA and protein production. For example, NINI, genomic copy, gains drive tumor growth and an immunologically sterile microenvironment in melanoma to increase transcription. The gene is transcribed at a higher rate due to stronger promoter activity or increased transcription factor binding. For example, mutations in a superenhancer upstream of talone can create MyB binding sites, resulting in the overexpression of talone in cancer. 3. Increased RNA stability. The nRNA is more stable and degrades more slowly, leading to increased protein production. 4. Increased translation. The nRNA is translated more efficiently, leading to increased protein production. Quantitative change. The amount of protein is increased, leading to a quantitative change in its function. Examples: HC-MyC overexpression in Burkitt's lymphoma due to translocation of c-MyC under the control of the immunoglobulin. Ig, promoter expression of HER2 in breast cancer due to gene amplification. In summary, gain-of-function mutations and overexpression are distinct mechanisms of oncogenesis. Gain-of-function mutations involve a qualitative change in protein function, while overexpression involves a quantitative increase in the amount of normal protein. Both can lead to an increased activity of signaling pathways and contribute to cancer development, but they do so through different means. Now, let's discuss how mutations in the Ra oncogene lead to uncontrolled cell proliferation, focusing on the mechanisms involved. Overview of RAS function. RAS proteins, which include HRAS and CRAS, are small GPases that act as molecular switches in cell signaling pathways. They cycle between an active GTP-bound state and an inactive GDP-bound state. In their active form, Ra proteins transmit signals that promote various cellular functions, including proliferation, survival, and differentiation. Mechanism of RAS activation. First, normal activation cycle. When a growth factor binds to its receptor on the cell surface, it activates guanine nucleotide exchange factors, GFs. These GFs facilitate the exchange of GDP for GTP on RAS, turning it on. Once activated, RAS interacts with downstream effectors such as RAF, part of the MEPK pathway, and PI3K, phosphoinositide 3 kinase, leading to signaling cascades that promote cell growth and survival GTPase activating proteins, GAPs, then stimulate ROS intrinsic GPase activity, hydrolyzing GTP back to GDP, and turning ROS off. Genetic mutations, ROS gene, particularly at codons 12, 13, or 61, led to a loss of function of GAPs or alter the intrinsic GPase activity of ROS. For example, a common mutation like CROS G12 prevents GTP hydrolysis. As a result, RA remains in its active, GTP-bound state for prolonged periods. Consequences of oncogenic ROS activation. 1. Sustained signaling. The inability to hydrolyze GTP means that oncogenic ROS continues to signal downstream pathways without regulation. This leads to constant activation of signaling cascades that promote cell proliferation and survival. The primary downstream effectors activated by oncogenic ROS include the MAPK pathway. This pathway regulates gene expression related to cell growth and division. Continuous activation can lead to increased transcription of genes that drive the cell cycle forward, and the PI3K-ACT pathway also. This pathway promotes cell survival by inhibiting apoptosis and enhancing cellular growth. Continuous activation can lead to resistance against cell death signals. Second cell cycle progression. Oncogenic ROS can also lead to hyperphosphorylation of the retinoblastoma protein, RB. When RB is phosphorylated, it releases ETWF transcription factors that promote the transcription of genes necessary for progression from the GON phase to the S phase of the cell cycle A. This transition allows cells to enter DNA synthesis and continue proliferating uncontrollably. Third, increased cell survival. By activating pathways that inhibit apoptosis, oncogenic RA promotes not only proliferation, but also survival of cells that would otherwise undergo programmed cell death. Fourth, genomic instability. Sustained activation of ROS can also contribute to genomic instability by promoting DNA damage response pathways or increasing reactive oxygen species ROS levels, further driving tumorigenesis and summary mutations in the ROS oncogene, lead to uncontrolled cell proliferation, primarily through sustained activation of signaling pathways due to impaired GTP hydrolysis. Oncogenic ROS remains in an active state, continuously signaling downstream effectors like MAPK and PI3K-ACT pathways that promote cell growth and survival while inhibiting apoptosis. This dysregulation is a key driver in many cancers, making ROS mutations critical targets for therapeutic intervention. The transfection assay is a powerful experimental technique used to identify oncogenes by introducing DNA from tumor cells into normal cells. This method allows researchers to observe the transformation of normal cells into cancerous cells, providing insights into the genetic factors that drive oncogenesis. Steps in the transfection assay. First, preparation of tumor DNA. The first step involves isolating genomic DNA from tumor cells, which may contain mutated or overexpressed oncogenes. This DNA can come from various sources, including fresh tumor biopsies or established cancer cell lines. Second, transfection into normal cells. The isolated tumor DNA is then introduced into normal recipient cells, often using a cell line, such as Ni3 mouse fibroblasts. This introduction can be achieved through various methods, including liposuction electroporation or chemical transfection reagents. 30. Selection and culture. After transfection, the cells are cultured under conditions that select for successfully transfected cells. This may involve using antibiotic selection if a selectable marker gene, like an antibiotic resistance gene, is included in the transfected DNA. Fourth, observation of foci formation. The key indicator of successful transformation is the formation of foci, clusters of transformed cells that exhibit altered morphology and growth characteristics compared to normal cells. These foci indicate that the introduced DNA contains oncogenic sequences capable of driving uncontrolled cell proliferation. Fifth, characterization of oncogenes. Once foci are identified, further analysis can be performed to characterize the specific oncogenes responsible for the transformation. This can include sequencing the integrated DNA or performing functional assays to confirm the presence and activity of known oncogenes. Example of oncogene discovery. A classic example of using the transfection assay for oncogene discovery involved the Eros family of oncogenes. Researchers transfected Ni33 cells with DNA extracted from human tumors and observed foci formation, leading to the identification of RAS, CRAS, and NRA as key oncogenes involved in cancer development. Advantages of the transfection assay. 1. Direct evidence. The assay provides direct evidence linking specific genetic alterations to cellular transformation. 2. Functional analysis. It allows for functional analysis of candidate oncogenes in a controlled environment. 3. Versatility. The method can be adapted to study various types of cancer by using different tumor DNA sources. Limitations. Incomplete representations. Not all oncogenes may be detected if they require specific cellular contexts or additional mutations for activation. Complexity of tumor genomes. Tumor genomes are complex, and identifying single transforming genes among many potential candidates can be challenging. In summary, the transfection assay is a crucial method in cancer research that enables the identification and characterization of oncogenes by introducing tumor-derived DNA into normal cells and observing their transformation into cancerous phenotypes. This technique has been instrumental in advancing our understanding of the genetic drivers of cancer. Now, however, let's investigate why the translocation of C-MYC to the immunoglobulin, e.g., locus causes lymphoma, specifically Burkitt's lymphoma. Burkitt's lymphoma is a type of aggressive B-cell non-Hodgkin lymphoma characterized by chromosomal translocations involving the NIC gene on chromosome 8 and 1 of the immunoglobulin, EGOCH, the heavy chain locus, I, on chromosome 14, in most cases or less, frequently the kappa light chain locus, IKE, on chromosome 2 or the lambda light chain locus, EGL, on chromosome 2020. Here's a detailed explanation of why this translocation leads to lymphoma. 1. Deregulation of MYC expression. EA-MYC gene encodes a transcription factor that regulates the expression of genes involved in cell growth, proliferation, and apoptosis. Normal MYC expression is tightly controlled, responding to growth signals and other stimuli. When C-MYC is translocated to the IG locus, it comes under the control of the strong IG promoters and enhancers, which are normally active in B cells to drive high levels of immunoglobulin gene expression. This results in constitutive high-level expression of MYC, independent of normal cellular signals. An enhancer, RNA, located at the I locus, has been shown to regulate MYC expression and control the proliferation and chemoresistance of Burkitt lymphoma cells with I-MYC translocation. 2D. Uncontrolled cell proliferation. The elevated levels of MYC protein drive uncontrolled cell proliferation. MYC activates the transcription of genes involved in cell cycle progression, DNA replication, and ribosome biogenesis, pushing cells through the cell cycle at an accelerated rate in Burkitt lymphoma cells with a T814 translocation. Activation of Epstein-Barr virus. Nuclear antigen. 2. EBNA2A induces growth arrest along with rapid repression of IgMu and c-MYC transcription. This indicates that c-MYC expression is crucial for maintaining the proliferation of these lymphoma cells. 3. Inhibition of apoptosis. MYC can also inhibit apoptosis, programmed cell death, by modulating the expression of genes involved in cell survival pathways. This allows the cells to accumulate and form tumors for genomic instability. epigenetics expression can lead to genomic instability, increasing the likelihood of additional mutations that further promote tumorigenesis. 5. Disruption of normal B-cell development. The deregulated MYC expression disrupts normal B-cell development and differentiation, leading to an accumulation of immature B cells that are prone to malignant transformation. The intronic enhancer, EA, is not physically associated with the MYC coding sequences after translocation and is, therefore, unlikely to be important for MYC deregulation. Elements in the I3Kp regulatory region are required for MYC translocation and/or deregulation. 6. Alternative mechanisms independent of MYC translocation. Some Burkitt lymphomas lack MYC translocation. In such cases, deregulation of microRNAs, miRNAs, that target c-MYC may be responsible for lymphomagenesis. In summary, the translocation of c-MYC to the Ig locus in Burkitt's lymphoma results in deregulated and high-level expression of the MYC oncogene driving uncontrolled cell proliferation, inhibiting apoptosis, promoting genomic instability, and disrupting normal B-cell development. This cascade of events leads to the development of aggressive B-cell lymphoma. Okay, let's explore how oncogenes such as ROS can epigenetically silence tumor suppressor genes contributing to cancer development. Epigenetic silencing refers to the inactivation of genes without altering their DNA sequence. This process often involves modifications to DNA or histone proteins that affect gene expression. Oncogenes can hijack these epigenetic mechanisms to silence tumor suppressor genes, further promoting cell proliferation, survival, and other hallmarks of cancer. Here's how oncogenes like Ra can epigenetically silence tumor suppressors. 1. Activation of epigenetic machinery. Oncogenes can activate or upregulate the expression of enzymes involved in epigenetic modifications, such as DNA, methyltransferases, DNMDs, histone deacetylases, HDAs, and histone methyltransferases, AMPTEs. For instance, activated ROS can induce the expression of DNMTone, an enzyme that adds methyl groups to cytosine bases in DNA, CpG methylation. 2. Recruitment of chromatin modifiers. Activated oncogenes can recruit chromatin-modifying enzymes to the promoter regions of tumor suppressor genus. This recruitment is often mediated by transcription factors or other regulatory proteins. In the case of ROS, studies have shown that activated ROS recruits DN-tone to the thyme-tone to the promoter region of the AES gene, a pro-apoptotic factor. This leads to DNA methylation of the FSAS promoter, 3. DNA methylation, and histone modification. ENA methylation, particularly at CpG islands in promoter regions, is a well-known mechanism of gene silencing. Methylated DNA recruits methyl binding domain, MBD, proteins which further repress transcription. In addition to DNA methylation, histone modifications can also contribute to gene silencing. For example, histone methylation, EEJ, H3K9Me3, or H3K27Me3, and histone decetylation are associated with repressed chromatin states. For example, one study demonstrated that long non-coding RNA part tone restrains aggressive gastric cancer through the epigenetic silencing of PDFB via the PLZF-mediated recruitment of H2O 4. Tumor suppressor. Gene silencing. The combination of DNA methylation and repressive histone modifications leads to chromatin condensation and transcriptional repression of tumor suppressor genes. In the case of RAS-mediated silencing of the methylation of the SF promoter prevents the binding of transcription factors, resulting in decreased FAS expression. This makes cancer cells more resistant to apoptosis. Another study has established that RAS GPase activating like protein, RASL, a cat wound-regulated RAS-GAP, is silenced through CpG methylation in multiple tumors. Ectopic expression of catalytically active RASL leads to growth inhibition of these tumor cells by RAS inactivation. 5. Maintenance of silenced state. Once a tumor suppressor gene is silenced, epigenetic marks can be maintained through multiple cell divisions, ensuring that the gene remains inactive in daughter cells. DNMTone, for example, is a maintenance methyltransferase that copies methylation patterns from the parental DNA strand of the newly synthesized strand during DNA replication. Examples and additional mechanisms. RASL silencing. In addition to the epigenetic silencing described in the present study, previous work has defined that downregulation of RASL can also be achieved in certain tumors through transcriptional control driven by PICI-MEX2. A study has identified methylcytosin guanine dinucleotide, CpG, binding protein 2, MESP2, as a novel oncogene. MEC2 resides in a region of the X chromosome that is significantly amplified across 18% of cancers, and many cancer cell lines have amplified, overexpressed MEC2, and are dependent on MESP2 expression for growth. BRAF. V600 mutations can lead to the epigenetic silencing of key genes, such as IGFBP7, that may act in concert with BRAF mutation to promote neoplasia. Donal. Disruptor of telomeric silencing 1-like modulates the senescence-associated secretory phenotype, SASP, through epigenetic regulation of ILNA-MIK. Ra-axis epigenetic alterations can impede epithelial mesenchymal transition by modulating centrosome amplification and MIK-RAS axis in triple-negative breast cancer cells. In summary, oncogenes like Ra can epigenetically silence tumor suppressor genes by activating epigenetic machinery, recruiting chromatin modifiers to tumor suppressor promoters, and inducing DNA methylation and repressive histone modifications. This leads to transcriptional repression of tumor suppressor genes promoting cancer development. Understanding these mechanisms is crucial for developing therapeutic strategies aimed at reversing epigenetic silencing and restoring tumor suppressor function. Okay, let's explore how gene amplification contributes to oncogene activation, drawing from the provided search results. Gene amplification is a mechanism by which the copy number of a specific DNA sequence is increased. This increase in gene dosage can lead to overexpression of the encoded protein, which in the case of oncogenes, can drive uncontrolled cell proliferation and contribute to cancer development. Here's a more detailed explanation. 1. Increased gene dosage. Gene amplification results in multiple copies of an oncogene within the cancer cell's genome. This increase in gene copy number directly leads to an increased template for transcription. 2. Overexpression of the oncogene. With more copies of the oncogene, the cell produces more nRNA transcripts, leading to increased translation and consequently higher levels of the oncogene protein. This overexpression can disrupt normal cellular processes and push the cell towards uncontrolled growth. The expression levels for genes within amplification. Hotspots are expressed at significantly higher levels with increased gene dosage. 3. Disruption of cellular signaling. Many oncogenes encode proteins involved in cell signaling pathways that regulate cell growth, division, and survival. When these oncogenes are overexpressed due to gene amplification, the signaling pathways become hyperactive, leading to uncontrolled cell proliferation and tumor formation. For example, multiple downstream components of the EVF family signaling pathway, including CDK5 acton and chacon, are overexpressed as a direct result of gene amplification in amplification. Lung cancer E4. Mechanisms of amplification. Gene amplification can occur through various mechanisms, including double-minute chromosomes, D9. These are extrachromosomal circular DNA fragments that contain amplified genes. Because they lack a centromere, they are often unevenly distributed during cell division, leading to heterogeneity in gene copy number among cells homogeneously staining. Regions, HSRs. These are abnormally expanded regions within a chromosome that contain amplified genes integrated into the chromosome structural breakage fusion bridge, BFB. BFB cycles. This mechanism involves cycles of chromosome breakage fusion and bridge formation during cell division, leading to increasing copy numbers of specific regions. The 1q12 pericentromeric heterochromatin region can serve as both a recurrent breakpoint as well as a fusion point for sister chromatids, which ultimately bracket both the proximal and distal boundaries of the amplicon. Examples of amplified oncogenes, ACE, MIC. Amplification of MIC is a well-known mechanism in various cancers. The sequence of a germline NMIC gene and amplification as a mechanism of activation has been reported. ERB2, HER2. Amplification of ERBD2 is frequently observed in breast cancer and is associated with increased cell proliferation and poor prognosis. EGFR. Amplification of EGFR is common in lung cancer and glioblastoma. MET. High-level MET amplification has also been reported as a resistance mechanism of rhosone tyrosine kinase inhibitors in rhosome rearranged non-small cell lung cancer. Increased protein expression often occurs without gene amplification because various mechanisms may cause an overexpression. Transcriptional upregulation induced by other oncogenes, environmental factors such as hypoxia or radiation, or substances. In summary, gene amplification is a significant mechanism of oncogene activation in cancer. By increasing the copy number of oncogenes, cells can produce elevated levels of oncogenic proteins, leading to dysregulation of cell signaling pathways and uncontrolled cell growth. This phenomenon has been observed in a variety of cancers and is a key target for therapeutic intervention. Chromothripsis is a fascinating and complex phenomenon that plays a significant role in cancer developments. It refers to a catastrophic event in which a chromosome undergoes massive fragmentation, resulting in numerous cluster genomic rearrangements, often localized to specific regions of one or a few chromosomes. This process can lead to the activation of oncogenes or the loss of tumor suppressor genes, thereby contributing to tumorigenesis. Mechanism of chromothripsis. 1. Catastrophic chromosomal shattering. Epichromothripsis typically occurs during a single catastrophic event in a cell's life cycle. This can happen due to various stressors such as ionizing radiation, replication stress, or errors during cell division. The most widely accepted model suggests that chromosomal fragmentation may occur in structures called micronuclei, which form as a result of mitotic errors. These micronuclei contain fragments of chromosomes that are not correctly segregated during cell division. 2. Error-prone repair. After fragmentation, the broken chromosome pieces are reassembled through error-prone repair mechanisms, primarily non-homologous end joining, ANI, or alternative end joining, ALT-EJ. These repair processes often lack fidelity and can lead to incorrect rejoining of chromosomal fragments. The reassembly may result in complex rearrangements, including deletions, duplications, inversions, and translocations. Contribution to cancer 1. Oncogene activation. Chromothripsis can lead to the amplification of oncogenes. For example, the MyC gene, which is a well-known oncogene involved in cell proliferation and growth, can become amplified through chromothripsis. This amplification results in overexpression of the MyC protein driving uncontrolled cell proliferation. Other oncogenes may also be activated through similar mechanisms of rearrangement and amplification due to chromothripsis to loss of tumor suppressor genes, in addition to activating oncogene. Chromothripsis can result in the loss or inactivation of tumor suppressor genes. The deletion of critical tumor suppressor genes can remove regulatory mechanisms that normally prevent uncontrolled cell growth, for instance. If chromothripsis leads to the deletion of genes such as T53 or RB, which are crucial for cell cycle regulation and apoptosis, this can further promote cancer development. 3. Genomic instability. The chaotic nature of chromothripsis contributes to genomic instability within cancer cells. This instability can lead to further mutations and alterations that enhance the aggressiveness of the tumor and contribute to treatment resistance. The presence of multiple rearrangements within the genome can create a diverse population of cancer cells with varying characteristics, making it more challenging for therapies to effectively target all tumor cells. Clinical implications. Chromothripsis has been observed in various types of cancers, including acute myeloid leukemia, AML, breast cancer, and other solid tumors. Its presence is often associated with poor prognosis due to the aggressive nature of tumors exhibiting this phenomenon. In summary, chromothripsis is a process characterized by catastrophic chromosomal shattering, followed by error-prone repair mechanisms that lead to complex genomic rearrangements. This phenomenon drives cancer by activating oncogenes like MyC through amplification and causing the loss of tumor suppressor genes. The resulting genomic instability contributes to tumor progression and treatment resistance, highlighting the importance of understanding chromothripsis in cancer biology and therapy development. Well, now, let's discuss the challenges of targeting ROS mutations in cancer therapy based on the provided search results. ROS mutations are among the most frequent oncogenic drivers in human cancers, found in approximately 30% of all tumor types. These mutations are crucial for tumor initiation, maintenance, and recurrence. For decades, ROS was considered undruggable. However, while there's been progress, significant challenges remain. One difficulty in direct targeting: lack of binding pockets. ROS proteins have a mostly flat surface and lack small molecule binding cavities, making it extremely challenging to develop drugs that bind directly and effectively. High affinity for GTP. ROS has a very high affinity for GTP, making it difficult for small molecules to compete and displace GTPa. 2. Mutation specificity, limited applicability. The recent approval of Cros12 inhibitors, such as adagrasib and sotorasib, has validated Cros as a direct therapeutic target. However, Cros12 is not a prevalent mutation in most cancer types, and these drugs exclusively inhibit this specific mutant. Other oncogenic mutants. Other ROS oncogenic mutants, such as G12, lack reactive cysteines, rendering them unsuitable for targeting via the same strategy. Resistance mechanisms. Adaptive resistance. Cancer cells can develop resistance to Cros inhibitors through various mechanisms, including primary adaptive and acquired resistance. These resistance mechanisms are being targeted in studies that combine Cros inhibitors with inhibitors of horizontal or vertical signaling pathways. ROS mutations drive resistance to targeted therapies, particularly FLT3, IDH2, or JAK2 inhibitors for complexity of ROS signaling. Multiple downstream effectors. ROS acts through many different pathways to elicit its tumor-promoting effects. This complexity and the potential for robust adaptive feedback continue to present substantial challenges. 5. Tumor microenvironment. Effects on the tumor microenvironment. Mutant CRAS has important effects on the tumor microenvironment, including the immunological milieu. These effects must be considered to fully understand resistance to CRAS inhibitors and when designing novel treatment strategies. Alternative signaling pathways. Pathway switching. Even when ROS is inhibited, cancer cells can activate alternative signaling pathways to maintain their growth and survival. 7 strategies to overcome challenges. Despite these challenges, new approaches have generated optimism about targeting specific ROS mutations in an allele-dependent manner. 4. Cancer therapy. Some of these strategies include protein engineering. Engineered proteins can recognize various surfaces with high affinity and specificity, offering a promising method to target ROS. Targeting ROS effector interactions, disrupting ROS dimerization, interrupting Ra nucleotide exchange, stimulating ROS interaction with tumor suppressor genes, promoting Ra degradation, combination therapies. Improvements in understanding ROS signaling in cancer cells and in the tumor microenvironment suggest the potential for several novel combination therapies, which are now being explored in clinical trials. In conclusion, targeting ROS mutations in cancer therapy is challenging due to the difficulty in direct targeting the diversity of ROS mutation, the development of resistance mechanisms, the complexity of ROS signaling, and the effects on the tumor microenvironment. However, ongoing research and the development of new therapeutic strategies offer hope for improving outcomes in patients with ROS mutant malignancies. Epigenetics. DNA methylation is a chemical modification where a methyl group, CHIC3, is added to a cytosine base in DNA, typically at cytosine guanine denucleotide sites, CPG sites. These CPG sites are often clustered in regions called CPG islands, which are commonly found in the promoter regions of genes. In normal cells, most CPG islands are unmethylated, allowing genes to be transcribed. However, in cancer cells, aberrant DNA methylation patterns emerge, leading to gene silencing. Here's a breakdown of the mechanism. 1. DNA methylation at CPG islands. In cancer cells, CPG islands in the promoter regions of tumor suppressor genes often become hypermethylated. This aberrant methylation is a well-established epigenetic deregulation in cancer. 2. DNMT enzymes. DNA methylation is catalyzed by a family of enzymes called DNA methyltransferases, DNMTs. The major DNMTs involved are AG-emmetone, functions as a maintenance methyltransferase, copying methylation patterns from the parental DNA strand to the newly synthesized strand during DNA replication. DNMT3D and DNT3B establish de novo methylation patterns and may also play a role in maintaining methylation. DNT biosubstrate activity is directed towards HA336 MA3 marked CPG islands. In tumor tissue, the expression of all 3 DNMT isoforms is significantly higher compared with matched normal-appearing tissue. 3-free. Recruitment of methyl-binding domain, MBD, proteins. Once CPG islands are methylated methyl CPG binding domain proteins to the methylated DNA. One such protein, MBT2, plays a key role in maintenance and spread of DNA methylation at CPG islands. 4. Chromatin remodeling and histone modification. MBD proteins recruit other chromatin-modifying enzymes, such as histone deacetylases, HDAC, and histone methyltransferases. This leads to histone deacetylation, removal of acetyl groups from histone tails resulting in a more condensed chromatin structure. Histone methylation, addition of methyl groups to histone tails, e.g. H3-9, IMA2, 3, or H3-27-3, which are associated with transcriptional repression. 5. Transcriptional repression, the combination of DNA methylation, MBD binding, and repressive histone modifications, results in chromatin condensation and transcriptional silencing of the associated gene. The unmethylated CpG islands of active genes are enriched in acetylated H3 and H4 and H3 form, 1-to-1. Nucleosomes are strongly positioned across CpG islands in their active and unmethylated state, except at the transcription start site, where there is often a 1-nucleosome gap. 6. Tumor suppressor gene silencing. When DNA methylation silences tumor suppressor genes, the normal regulatory mechanisms that control cell growth and prevent tumor formation are disrupted. This can promote uncontrolled cell proliferation, survival, and other hallmarks of cancer. In summary, DNA methylation at CpG islands contributes to gene silencing in cancer by recruiting MBD proteins and chromatin-modifying enzymes, leading to histone deacetylation, histone methylation, chromatin condensation, and transcriptional repression of tumor suppressor gene S. This process disrupts normal cellular regulation and promotes cancer development. General transcription. Factor binding at CpG islands in normal cells correlates with resistance to de novo DNA methylation in cancer cells. Okay, let's compare southern blot and basalphite sequencing for methylation analysis drawing from the provided search results. Both techniques are used to study DNA methylation, but they differ significantly in their methodology, resolution, and applications. 1. Southern blot principle. Southern blotting is a technique used to detect specific DNA sequences and can be adapted to determine DNA methylation status. The method relies on digesting genomic DNA with restriction enzymes that are sensitive to methylation procedure. Genomic DNA is digested with methylation-sensitive restriction enzymes. These enzymes can't cut DNA if their recognition sequence is methylated. The digested fragments are separated by size, using agarose gel electrophoresis, and then transferred to a membrane. Southern blot, the DNA on the membrane, is hybridized with a labeled probe, allowing for detection. The resulting band patterns are analyzed detection. The probe is typically labeled either radioactively or non-radioactively for detection. Non-radioactive methods involve using a fluorescence-labeled antibody resolution. Southern blotting provides an overall estimate of methylation within the region recognized by the restriction enzyme. It does not offer single base resolution. It is limited to CGS that occur in a DNA sequence context that is recognized by a methyl-sensitive restriction enzyme. Advantages: can assess large repeat expansions or contractions, such as those in triplet repeat disorders, useful for determining if triplet repeat expansions lead to methylation of the affected region. Robust, reproducible, and relatively simple. Disadvantages: requires relatively large amounts of genomic DNA, limited to CGS, that occur in a DNA sequence context that is recognized by a methyl-sensitive restriction enzyme. Tedious, low throughput, and provides inexact sizing across all categories of allelase time-consuming applications. Southern blotting is particularly useful in the molecular genetic examination of triplet repeat disorders, such as Fragile X syndrome, where it helps determine if triplet repeat expansions lead to methylation of the affected region. Southern blot and DNA labeling detection by double DNA digestion with Akori and Igi reveals both the presence of the mutation and the methylation status. 2. Bisulfite sequencing principle. Bisulfite sequencing provides single-base resolution of DNA methylation patterns. The method relies on treating DNA with sodium bisulfite, which converts unmethylated cytosines to uracil, while methylated cytosines remain unchanged. Procedure remain unchanged. DNA is treated with sodium bisulfite, deaminating unmethylated cytosine to uracil. The bisulfite-converted DNA is then amplified using PCR. The PCR products are sequenced. The resulting sequence is compared to the original sequence to determine the location of methylated cytosines detection. Sequencing allows for the direct identification of methylated cytosines at single-base resolution. Bisulfite sequencing offers single-base resolution, providing detailed information on the methylation status of individual CpG sites. Advantages: provides single-base resolution of DNA methylation patterns, can analyze methylation at every cytosine in a region of interest. Disadvantages: more complex and time-consuming than Southern blotting, requires specialized equipment and expertise. Applications: bisulfite sequencing is used to detect the methylation status of individual CpG sites in the entire promoter region. Okay, now, let's explore the link between Mulhone methylation and microsatellite instability, MSI, drawing from the provided search results. Mulhone is a key gene in the mismatch repair, MMR, system, which is responsible for correcting errors that occur during DNA replication. Silencing of Mulhone due to promoter methylation leads to a dysfunctional MMR system, resulting in microsatellite instability. Microsatellites are short, repetitive DNA sequences that are prone to replication errors. Here's a detailed breakdown of why Mulhone methylation leads to MSI. 1. Mulhone and mismatch repair, MRON, encodes a protein that forms a heterodimer with PMS2, and this complex is essential for MR function. The MR system normally identifies and repairs base-base-base mismatches and insertion-deletion loops that occur during DNA replication. 2. Mulhone promoter methylation. Methylation of the Mulhone promoter is an epigenetic modification that leads to gene silencing. Specifically, hypermethylation prevents the normal activation of the DNA repair gene. This methylation is a frequent event in endometrial cancer. 3. Silencing of Mulhone expression. When the Mulhone promoter is methylated, it disrupts the transcription of the Mulhone gene, resulting in reduced or absent Mulhone protein production. This silencing stops the formation of Mulhone protein. 4. Impaired mismatch repair. Without functional Mulhone protein, the MMR system is impaired. This prevents the normal activation of the DNA repair gene. The Mulhone promoter hypermethylation is an important event silencing the Mulhone gene expression and preventing the formation of Mulhone protein and normal activation of the DNA repair gene. 5. Microsatellite instability, MSI. With a defective MR system, errors in microsatellite regions accumulate because they are no longer corrected. This accumulation of errors leads to microsatellite instability, MSI, characterized by changes in the length of microsatellite sequences compared to normal tissue. MSI is involved in the genesis of about 15% of sporadic colorectal cancers, CRCs. 6 consequences of MSI. MSI can result in frameshift mutations in coding regions, leading to the production of non-functional proteins. MSI is associated with distinct clinicopathological features and can influence the response to chemotherapy. Tumors with a high frequency of microsatellite instability tend to be diploid, to possess a mucinous histology, and to have a surrounding lymphoid reaction. MSI is a hallmark of Lynch syndrome, a hereditary cancer syndrome caused by germline mutations in MMR genus 7 Milhone methylation in cancer. Milhone promoter methylation is frequently observed in sporadic colorectal cancers and endometrial cancers with high microsatellite instability, MSIH. It is an important cause for genomic instability and cell proliferation to the point of colorectal cancer formation. In summary, Milhone methylation is linked to microsatellite instability because methylation of the Milhone promoter silences Milhone gene expression, disrupting the mismatch repair system and leading to the accumulation of errors in microsatellite regions. This results in the MSI phenotype, which has significant implications for cancer development, diagnosis, and treatment. MyC and ROS are two of the most frequently activated oncogenes in cancer, and their cooperation plays a significant role in driving cancer progression. Here is how they work together to enhance tumorigenesis. Mechanisms of cooperation. 1. Synergistic effects on cell proliferation. MyC is a transcription factor that regulates the expression of genes involved in cell growth, proliferation, and metabolism. It drives the cell cycle forward by promoting the transcription of genes that facilitate progression through the GON phase and into S phase. RAS, when activated, initiates signaling cascades that promote cell division and survival primarily through pathways such as the MAPCAM, PI3K, ACT pathways. When MyC and ROS are co-activated, they synergistically enhance cellular proliferation beyond what either can achieve alone. 2. Regulation of apoptosis. RAS has been shown to have dual roles in apoptosis regulation. While it can promote survival signals, it can also activate pro-apoptotic pathways under certain conditions. MyC, on the other hand, is known to induce apoptosis if not properly regulated. The cooperation between MyC and ROS helps mitigate the apoptotic effects that might induce, allowing cells to survive despite high levels of MyC expression. This is particularly important in cancer cells where evasion of apoptosis is a hallmark of tumorigenesis. 3. Stabilization of MyC protein. ROS signaling can enhance the stability of MyC protein by promoting its phosphorylation at specific residues, e.g. SIR62. This phosphorylation prevents MyC from being targeted for degradation, allowing it to accumulate and exert its oncogenic effects more effectively. By stabilizing MyC, RA not only boosts MyC's transcriptional activity, but also amplifies its ability to drive cell growth and proliferation. 4. Influence on tumor microenvironment. Recent studies have shown that MyC and ROS cooperate to modulate the tumor microenvironment. They can influence immune responses by regulating immune checkpoint molecules and cytokine production. For example, ROS activation has been linked to changes in immune cell recruitment and activity within the tumor microenvironment, which can further support tumor growth and survival. 5. Context-dependent cooperation. The nature of cooperation between MyC and ROS can vary, depending on the specific cancer type and genetic context. In some tumors, they may work through overlapping mechanisms to drive tumorigenesis, while in others, they may function through distinct pathways. For instance, in hematological malignancies, MyC is often more potent than RAS at initiating tumors. However, their combined activation accelerates tumor development. In breast cancer, mutations in RAS can sometimes compensate for loss of MyC function. Clinical implications. The cooperation between MyC and ROS presents both challenges and ROS opportunities for cancer therapy. Targeting pathways. Understanding how these oncogenes interact provides insights into potential therapeutic targets. For instance, targeting downstream effectors of both ROS and MISC could be a strategy to inhibit their cooperative effects. Combination therapies designed to disrupt their cooperation or target both pathways may enhance treatment efficacy and overcome resistance mechanisms. In summary, MIC and ROS cooperate to drive cancer progression through synergistic effects on cell proliferation, regulation of apoptosis, stabilization of each other's protein products, modulation of the tumor microenvironment, and context-dependent interactions. This cooperation underscores the complexity of oncogenic signaling networks in cancer and highlights potential avenues for therapeutic intervention. Okay, let's describe Knutson's two-hit model using the retinoblastoma RB gene as an example. Knutson's two-hit hypothesis, also known as the Knutson hypothesis, explains how tumor suppressor genes like RB contribute to cancer development. It proposes that both alleles of a tumor suppressor gene must be inactivated to cause a phenotypic change. This hypothesis was first formulated by Alfred G. Knutson in 1971, based on his statistical analysis of retinoblastoma, a malignant tumor of the retina. Here is a breakdown of the model using RB as an example. The ERB gene, the Arbone gene, is a tumor suppressor gene located on chromosome 13 that codes for a protein, PB, regulating the cell cycle. The protein inhibits the cell cycle, preventing cells from dividing uncontrollably. Inactivation of both copies of the Arbone gene leads to uncontrolled cell division and the development of retinoblastoma, two-hit hypothesis. Knudsen suggested that 2 hits to DNA were necessary to cause the cancer. An inactivation of both alleles is required as a single functional tumor suppressor. Gene is usually sufficient. Inherited retinoblastoma. In children with inherited retinoblastoma, one mutation in the Arbone gene is inherited from a parent. This means that every cell in their body already carries 1 hit. The second hit, which inactivates the remaining functional Arbone allele, occurs in a somatic retinal cell. Because only 1 additional mutation is required, inherited retinoblastoma tends to develop H1 earlier in life, too often in both eyes, bilateral EAS3, with multiple tumors in each eye, sporadic retinoblastoma. In non-inherited sporadic retinoblastoma, an individual is born with 2 functional copies of the Arbone gene in all their cells. In this case, both hits must occur somatically in the same retinal cell. This means that 2 independent mutations inactivating both Arbone alleles must occur in the same cell. Because 2 mutations are required in the same cell, sporadic retinoblastoma tends to develop each one later in life. 2 in only 1 eye, unilateral, with a single tumor in the EA, loss of heterozygosity. Low. The second hit often involves loss of heterozygosity, low, where the normal Arbone allele is lost and the mutated allele is duplicated. This can occur through various mechanisms such as chromosomal deletion, mitotic recombination, or gene conversion. In summary, Knutson's 2-hit model explains the development of retinoblastoma based on the inactivation of the Arbone tumor suppressor gene. In inherited cases, 1 mutation is inherited and a second somatic mutation is sufficient to cause cancer. In sporadic cases, 2 somatic mutations must occur in the same cell. This model highlights the importance of both genetic and somatic mutations in cancer development and has been instrumental in understanding the role of tumor suppressor genes in cancer. Okay, let's dive into how RB phosphorylation regulates the cell cycle, drawing from the provided search results. The retinoblastoma protein, RB, encoded by the Arbone gene, is a crucial regulator of the cell cycle, particularly the transition from the GON phase to the S phase. RB's primary function involves binding to EDOF transcription factors, thereby controlling the expression of genes necessary for cell cycle progression. Phosphorylation of RB disrupts this interaction, influencing cell proliferation. Here's a breakdown of how RB phosphorylation regulates the cell cycle, RB's role in growth arrest. In its hypophosphorylated state, RB binds to EDOF transcription factors, preventing them from activating the transcription of their target genes. These target genes include those involved in DNA replication, cell cycle progression, and cell growth. By binding to EDOF, RB effectively blocks the cell cycle at the GON-S transition, preventing cells from entering S phase and undergoing DNA replication. Also, RB interacts with histone deacetylases, HDAC, 1, 2, and 3 histone methylase, SUVI39A1 and chromotyling enzymes, Bragon and BAR-MN, thus repressing cyclin CDK complexes and RB phosphorylation. The phosphorylation of RB is mediated by cyclin-dependent kinases, CDKs, in complex with cyclins. During the cell cycle, different cyclin CDK complexes are activated at specific times to regulate RB phosphorylation. In early to mid-GON, D-type cyclins, cyclin D, form complexes with CDK4 and CDK6. These complexes initiate the phosphorylation of RB. In late GON cyclins, E and A form complexes with CDKT. These complexes continue to phosphorylate RB, leading to hyperphosphorylation. CDK5, Actona, and Chacone are overexpressed as a direct result of gene amplification in lung cancer release of EDOF transcription factors. When RB is phosphorylated by cyclin CDK complexes, it undergoes a conformational change that reduces its affinity for EDOF transcription factors. This causes EDOF to be released from RB, allowing EDOF to activate the transcription of its target gene's progression into S phase. The activation of EDOF target genes promotes the entry of the cell into the S phase, where DNA replication occurs. These genes encode proteins necessary for DNA synthesis, such as DNA polymerase, as well as proteins involved in cell cycle progression, regulation by phosphatases. The phosphorylation status of RB is tightly regulated by the balance between CDK activity and phosphatase activity. Protein phosphatases, such as PON and PP2OA, dephosphorylate RB, returning it to its hypophosphorylated state and allowing it to inhibit EDOF activity. ATM's role. ATM regulates the cell cycle through control of PRB phosphorylation. In summary, RB phosphorylation regulates the cell cycle by controlling the activity of EDOF transcription factors. In its hypophosphorylated state, RB binds to EDOF and blocks the expression of genes required for cell cycle progression. Phosphorylation of RB by cyclin CDK complexes releases EDOF, allowing cells to enter S phase and undergo DNA replication. The dynamic balance between phosphorylation and dephosphorylation of RB is essential for proper cell cycle control. Disruption of this balance, such as through overexpression of cyclins or inactivation of RB, can lead to unchecked cell proliferation in cancer. Okay, let's examine why mutations in the 53 gene lead to genomic instability based on the search results. 53 is a critical tumor suppressor gene that plays a central role in maintaining genomic stability. It responds to various cellular stresses, including DNA damage, hypoxia, and oncogene activation. Mutations in 53 disrupt its normal functions, leading to genomic instability and increasing the risk of cancer development. Here's a breakdown of how 53 mutations lead to genomic instability. 1. Impaired DNA damage response. Wild-type 53 induces cell cycle arrest. allowing cells time to repair damaged DNA and preventing further proliferation of damaged cells, thus avoiding the propagation of mutations. In response to DNA damage, 53 activates the transcription of genes involved in cell cycle arrest, DNA repair, and apoptosis. Mutations in 53 impair its ability to activate these genes, compromising the DNA damage response. 2 defective cell cycle checkpoints. 53 is crucial for cell cycle checkpoint control. When DNA damage occurs, 53 induces cell cycle arrest, giving the cell time to repair the damage before replication. Mutations in 53 disrupt these checkpoint responses, allowing cells with damaged DNA to continue dividing. This leads to the accumulation of genetic errors and genomic instability. 3. Reduced apoptosis. 53 can trigger apoptosis, programmed cell death, in cells with irreparable DNA damage. This prevents the proliferation of cells with significant genetic abnormalities. Mutations in 53 can impair its ability to induce apoptosis, allowing cells with damaged DNA to survive and propagate, contributing to genomic instability. 4. Dysregulation of DNA repair mechanisms. 53 regulates genes involved in DNA repair pathways. Disruption of 53 function can impair DNA repair mechanisms, leading to the accumulation of DNA damage and genomic instability. For example, 53 coordinates base excision repair (BARE) to prevent genomic instability. Impaired 53 function leads to a failure of the BARE coordination mechanism and results in genomic instability. 5. Increased chromosomal aberrations. 53 mutations are associated with increased chromosomal aberrations, such as aneuploidy chromosomal rearrangements and chromothripsis. These aberrations contribute to genomic instability and can drive cancer development. Errors in mitosis can result in chromosome misaggregation, chromosomal rearrangements, and aneuploidy, which are main cause of genomic evolution of cancers. 6. Epigenetic dysregulation. 53 has a direct impact on site-specific epigenetic regulations and can physically interact with DNA and histone methyltransferases. By directly binding to DNA methyltransferases or indirectly affecting their expression, 53 can promote DNA demethylation or inhibit de novo DNA methylation at specific genomic loci. 7. Link to genomic instability and other genes. Loss of TP53 can lead to an inability to increase expression of NMYC down-regulated gene 1, endrigon, which leads to supernumerary centrosome formation, a known mechanism of aneuploidy. In summary, mutations in the 53 gene led to genomic instability by impairing the DNA damage response, disrupting cell cycle checkpoints, reducing apoptosis, dysregulating DNA repair mechanisms, and increasing chromosomal aberrations. This genomic instability promotes cancer development by allowing cells with damaged DNA to survive and proliferate, accumulating further genetic errors. Defects in mismatch repair, EMR, contribute to cancer through several mechanisms that lead to genomic instability and the accumulation of mutations. EMR is a crucial DNA repair pathway responsible for correcting base-base mismatches and insertion-deletion loops that occur during DNA replication. Here's how MMR defects drive cancer. 1. Accumulation of mutations. MMR defects result in a mutator phenotype characterized by an increased mutation rate. When the MR system is compromised, errors that occur during DNA replication are not corrected, leading to the accumulation of mutations throughout the genome. This accumulation can affect both proto-oncogenes and tumor suppressor genes, contributing to oncogenesis. 2. Microsatellite instability, MSI. One of the hallmark features of MMR deficiency is microsatellite instability, MSI. Microsatellites are short, repetitive DNA sequences that are particularly prone to replication errors. In MR deficient cells, these errors lead to changes in the length of microsatellite regions due to insertions or deletions, resulting in MSIs. Tumors exhibiting high levels of MSI are often associated with specific types of cancer, such as colorectal cancer and endometrial cancer. 3. Activation of oncogenes. The accumulation of mutations can lead to the activation of oncogenes, which drive uncontrolled cell proliferation. For example, mutations in genes that regulate cell growth and division can push cells toward a malignant phenotype. 4. Inactivation of tumor suppressor genes. Conversely, MMR defects can also result in the inactivation of tumor suppressor genes. Mutations that disrupt the function of these genes remove critical regulatory mechanisms that normally prevent uncontrolled cell growth. For example, mutations in the TP53 gene can lead to loss of cell cycle control and apoptosis. 5. Genomic instability. The inability to correct DNA replication errors leads to widespread genomic instability, which is a key characteristic of cancer cells. This instability can result in chromosomal rearrangements, aneuploidy, abnormal number of chromosomes, and further genetic alterations that promote tumorigenesis. 6. Hereditary non-polyposis colorectal cancer, NPCC. Defects in MMR are notably linked to hereditary non-polyposis colorectal cancer, NPCC, also known as Lynch syndrome. Individuals with germline mutations in MMR genes, such as MHON, MSTU, have an increased risk of developing various cancers at an early age due to the inherited predisposition for MSI and subsequent genomic instability. 7. Resistance to therapy. MMR-deficient tumors often show resistance to certain chemotherapeutic agents, such as alkylating agents and some forms of immunotherapy. This resistance is partly due to the high mutational burden that can lead to the generation of neoantigens, making these tumors more responsive to immune checkpoint inhibitors. In summary, defects in mismatch repair contribute to cancer by allowing the accumulation of mutations, leading to microsatellite instability and genomic instability. These changes promote oncogene activation and tumor suppressor gene inactivation, driving tumorigenesis. Understanding these mechanisms is crucial for developing targeted therapies and improving treatment outcomes for patients with MR-deficient tumors. Okay, let's outline the genetic steps in colorectal cancer development using the information from the provided search results. Colorectal cancer, CRC. Development is a multi-step process involving the accumulation of genetic and epigenetic alterations that transform normal colorectal epithelium into an adenoma and ultimately into an invasive and metastatic carcinoma. This progression, known as the adenoma-carcinoma sequence, is a widely accepted model for CRC development. Though colorectal tumors are heterogeneous at a genetic level, they appear to develop via several distinct pathways. The pathways of chromosome instability, SIN, microsatellite instability, and serrated neoplasia provide some information about their mechanisms of pathogenesis. Here's a step-by-step outline of the genetic events involved. 1. Initiation. APC loss inactivation. The initiating event in the SIN pathway is often the loss or inactivation of the APC, adenomatous polyposis coli, gene on chromosome 5q. APC is a tumor suppressor gene that regulates the WNT signaling pathway. Loss of APC function leads to the accumulation of beta-catenin in the nucleus, which then activates the WNT signaling pathway, resulting in increased cell proliferation. In tumors without APC mutation, there is often a mutation in the beta-catenin gene, CTN bone. This is considered an early event in colorectal tumorigenesis. 2. Proliferation. Cross-activation. Following APC loss, activating mutations in the cross-oncogene are commonly observed. Cross encodes a small GPase involved in cell signaling pathways that control cell growth and differentiation. Mutations in CRAS result in constitutive activation of the RS protein promoting cell growth and progression of adenoma. 3. Progression. Loss of tumor suppressor genes. As the adenoma progresses, additional genetic alterations occur, including the loss of other tumor suppressor genes such as T53 on chromosome 17p and DCC, deleted in colorectal carcinoma on chromosome 18q. Loss of T53 function impairs DNA repair and cell cycle control, allowing cells with damaged DNA to survive and proliferate. 4. Other mutations. Mutations involving other genes have been described, such as the TGFBR and PIK3CA. 5. Metastasis. Further genetic changes contribute to the development of invasive and metastatic carcinoma. 6. Additional pathways and factors. While the adenoma carcinoma sequence is a key pathway, other pathways such as the microsatellite instability (MSI) pathway and the serrated pathway also contribute to CRC development. Epigenetic alterations, such as aberrant methylation of tumor suppressor genes, also play a role in CRC. Genetic variations, acquired factors, and anatomical site-specific impacts can affect CRC development. In summary, colorectal cancer development is a multi-step process, driven by the accumulation of genetic and epigenetic alterations. The adenoma carcinoma sequence typically involves APC loss, CRAS activation, and loss of TP53 and DCC, leading to uncontrolled cell proliferation and tumor progression. Understanding these genetic steps is crucial for developing effective strategies for CRC, prevention, diagnosis, and treatment. DNA hypomethylation and hypermethylation are two distinct epigenetic modifications that, when acting in concert, can significantly contribute to cancer development. These alterations disrupt normal gene expression patterns, leading to the activation of oncogenes and silencing of tumor suppressor genes, TSGS. 1. Genome-wide hypomethylation mechanism. Genome-wide hypomethylation involves a global reduction in DNA methylation levels across the genome. This can lead to the activation of oncogenes by increasing their transcriptional activity. Oncogene activation. Hypomethylation can activate oncogenes such as CRAS. This activation results in increased expression of proteins that drive cell proliferation, survival, and other hallmarks of cancer. Localized hypermethylation mechanism. Localized hypermethylation involves increased DNA methylation at specific regions, particularly CPG islands in the promoter regions of genes. This leads to transcriptional silencing of the affected genes. Tumor suppressor gene silencing. Hypermethylation often silences tumor suppressor genes such as MLH and APC. This silencing removes critical regulatory mechanisms that normally control cell growth, DNA repair, and apoptosis, facilitating uncontrolled cell proliferation and tumor formation. For example, MLH promoter methylation in colorectal cancer leads to microsatellite instability. 3. Cooperation between hypomethylation and hypermethylation. Disruption of gene expression. Hypomethylation and hypermethylation cooperate to create an environment where oncogenes are activated and tumor suppressor genes are silenced. This dual effect accelerates tumorigenesis by promoting cell proliferation and inhibiting normal cellular regulation. Genomic instability. Global DNA hypomethylation is linked to genomic instability. Hypomethylation at centromeric. Repeat sequences can lead to chromosomal instability, increasing the likelihood of mutations and chromosomal aberrations. Epigenetic, genetic interplay. Epigenetic alterations can induce genetic changes and contribute to tumor progression. For instance, epigenetic silencing of secreted frizzled-related proteins SFRPs, induces abnormal activation of the WNT signaling pathway, further promoting cell proliferation. As a result of survival and proliferative advantages, these cells accumulate genetic mutations in other components of the WNT signaling pathway heterochromatin protein 1, HPONMUA, HEOUAD. Epigenetic and genetic mechanisms intertwine and take advantage of each other during tumorigenesis. Epigenetics cooperate with genetics and cell-to-cell signaling in defining cancer cell behavior at various stages of the disease. In summary, DNA hypomethylation and hypermethylation jointly drive cancer by activating oncogenes and silencing tumor suppressor genes, leading to genomic instability and disrupted cellular regulation. This cooperation between epigenetic and genetic mechanisms underscores the complexity of cancer development